Compared with other methods, this protocol is easier to find out new antagonists, agonists, and allosteric modulators quantitatively and qualitatively, without knowing the high-resolution structural information of the target protein. This protocol is highly efficient, easy to handle, economical, and commercially available. Combine this high-throughput screening method with ITC analysis. The functional small peptides can be obtained quantitatively and qualitatively. Demonstrating the procedure will be Ying Zhao and Qiang Wang, graduate students from our laboratory.
To begin this procedure, prepare a solution of FGFR2 encoding buffer at a concentration of 10 micrograms per milliliter. Add 1 milliliter of this solution to a 35 square centimeter dish and swirl repeatedly until the surface is completely wet. Incubate overnight at 4 degrees Celsius with shaking. The next day, pour off the coating solution and add the prepared blocking solution. Incubate at 4 degrees Celsius for at least 1 hour. Then, discard the blocking solution and quickly wash the plate 6 times with TBST while making sure the dish does not dry out. Reconstitute the original phage library in 1 milliliter of TBST and add this to the coated dish. Gently rock the plate on a shaker at room temperature for 2 hours. After this, discard the supernatant and wash the dish 10 times with TBST.